Hi, everyone. First of all, I would like to introduce myself. My name is Penny Ahmadi, and in this opportunity, I would like to talk a little bit about my research with the title Cancer Therapy by a Pro-Apoptotic Peptide. In this talk, I'm going to introduce in vivo therapeutic chemistry, which is developed in Professor Katsunori Tanaka's lab in Riken, Japan. But before that, I would like to talk a little bit about the cancer treatments. As we know that there are a lot of cancer treatment have been developed in the last decades, such as chemotherapy, radiotherapy, immunotherapy, hormone therapy, targeted therapy, and many more. However, from those reported cancer treatments show severe side effect. Even the side effect itself is more dangerous than the cancer cells. For example, the blood clotting. As you know, that blood clotting is very dangerous because it may cause a sudden death. With those side effects, we come up with a new strategy that we can eliminate the side effect. So we want to do a synthesis in the living cells. So the strategy is here. So basically, we need two important components that we want to use in this strategy. So first, is a catalyst, or we can combine it with a targeting agent, so we can call it metalloenzyme. And then the second thing is the substrate of the product itself. It means that the product is not active at all. At first, we inject intravenously the metalloenzyme to the human or patients or in mice. Then this metalloenzyme will be carried by blood and circulated into the whole body, and finally will accumulate it in a tumor. And after some time, we inject the second compound. It's a prodrug or substrate, and intravenously to the human. And then after that, it will be carried by blood, and also it will circulate in the whole body. And whenever the substrate of the prodrug meet the metalloenzyme in a tumor, this prodrug can be activated to be a drug and can kill the cancer cells. So therefore, this strategy is really promising because it may have several advantages, such as selective and specific, effective and efficient, and also the most important thing that it can reduce off-targeting problem. To do in vivo therapeutic chemistry, we need some reactions in vivo that we need to modify the drug from active to inactive, and then after that we want to activate again the prodrug. So here basically the structures of the drug contain amine. In this time we want to modify the amine moiety, and to modify this amine moiety and to make it inactive, we can attach the side chain with the alloc moiety or propargyl amine moiety, or with the propargyl ester moiety or another propargyl ester moiety. So this is one of the example of the reactions in the chemistry that we can use. And this reaction is so-called bioorthogonal chemistry because we want to do these reactions inside the living cell, and this reaction should not interfere native bioreactions in human or in living things. And before we need to prove, is it possible to do reactions in vivo? So here we did cell-specific, organ-specific labeling by using our chemistry. So here we use gold as a catalyst, and we combine it with specific targeting. In this case, we use a glycan. And depending on the glycocluster that we use, if we use metalloenzyme with the sialic acid as a targeting agent, this metalloenzyme can be specifically delivered to the liver. And when we use galactose, we can deliver this metalloenzyme specifically to the intestine. Then after this metalloenzyme accumulated in a specific organ, we try to inject intravenously fluorescence propargyl esters. And whenever this propargyl ester meet the gold in a specific organ, the triple bond will make a coordinate bond with the gold, and then nucleophilic attack on the surface of the organ will attack carbonyl and form amide bond. And then this specific organ can be labeled by our fluorescence. And here's the in vivo imaging that we got. As you can see here, here is the control that you can see no fluorescence were observed here. And then in the presence of the sialic acid as a targeting agent, you can see fluorescence intensity in the liver. And when we use galactose, we can see the fluorescence intensity in the intestine. So therefore, it is very possible to do in vivo reactions. And it's really promising for us. And then this result has been published in the Angewandte Chemical International Edition in 2017. For more detail, you can go and check for our paper. And then now we want to improve our chemistry, not only by changing or attaching side chain in the amine moiety, but in this case, we want to do another reaction, so-called ring closing metathesis reactions. So first, we have a natural product here. As you know, that there are a lot of structures of natural product, and it's really active, and the structure itself is really unique and complicated. And in this case, we want to open the ring to inactivate the drug. So here is natural product with a cyclic ring as a standard, as a model, and then we inactivate the product by opening the ring, and then after that we want to cyclize again this product inside the living things by using our metalloenzyme. In this case, we want to use ruthenium as a catalyst. And finally, this reaction, so we want to apply it to the in vivo therapy. And here is the model. So we choose a lot of natural product with the ring cyclic in his structures, and we want to modify the structures. And by opening the ring, we want to get inactive form of the natural product. So here we choose a very simple compound. It is umbelliprenine. It was isolated from plant Amimagus L, and it has activity as an anti-cancer too. And actually, we try many natural products, especially from marine organism, but actually from marine organism, the structure is really unique and complicated. So it is very difficult for us to use this as a first trial. So here we use umbelliprenine as the first model. As you can see here, the structure is really simple, and you have a ring and heat, and we can open the ring in a coumarin moieties. And after that, after we synthesize the product by opening the ring of the coumarin, and we check the cytotoxicity of these two compounds against HeLa-S3 cervical cancers, and also F549 lung cancers, and also SW620 colon cancers. And from this result, you can see that the different activity with prodrug and umbelliprenine is really significant, especially in a SW620 case. As you can see here, that prodrug almost no activity at all compared to the umbelliprenine. So they have a very significant difference between umbelliprenine and in its prodrug. So therefore, we use this umbelliprenine as a really perfect starting material for this strategy. And here, the product did not show any activity. So therefore, we use prodrug of umbelliprenine as a starting material, and we want to apply it for further analysis. Before we did reactions in vitro or in vivo, we need to confirm that our metalloenzyme can really specifically target organs. So here we modified our metalloenzyme with several glycan. So depending on the glycan that we use, we can deliver this metalloenzyme to the specific organs such as liver, intestine, or even a tumor. And after modifying the metalloenzyme with the glycan, we are ready for next experiments. So the reaction is here. So we tried to do reactions in vitro at the first time, because it's really need to confirm, is it really active against cancer cell or not? So here we treated this SW620 cell line with a prodrug in the presence of the metalloenzyme, in this case, ruthenium. As you can see, here is the result. So this is untreated cells, and this is a prodrug. The prodrug did not have any activity, and when we treat a prodrug in the presence of the ruthenium, the tumor growth can be, the cell viability can be suppressed here. And this the activity as strong as the umbelliprenine itself. So it means that our reaction is occurred in vitro. So therefore, this strategy is really promising to be conducted in further purposes, such as in vivo therapy. And actually, this result has been published in Nature Catalysis in 2019. For more detail, you can go and check for our paper for sure. And next, what we want to do is that we have specific organ labeling, and then we try to do reactions, then it is possible. And with those strategy in hands, we want to try our chemistry for more advanced purposes, such as organ-specific tagging to treat cancers. So the strategy is called selective cell tagging strategy, or SECT. So strategies here, this is the basic strategy that we want to apply for our chemistry. So here it's W620 colon cancers. As you know, that cancer is overexpressing integrin. And here we use RGD as a tumor targeting instead of the glycan. As you know that RGD have a strong interaction to the integrin. So therefore, we expect that our metalloenzyme can be specifically delivered to the tumor cells. And after the metalloenzyme accumulated in the tumor, we treat the tumor with the peptide propargyl esters. And as you know, as I mentioned before with our first strategy, that whenever there's triple bond with this metalloenzyme, they will make a coordinate bond with the gold, and then nucleophilic attack from amine moiety on the surface of the cancer cell will attack to the carbonyl and then form amide bond. And then this cell will be attacked by our peptide and can cause apoptosis, and it will cause the cell death. And here is the therapeutic in vivo synthetic chemistry by using SECT strategy. First, the important thing that you need to prepare is the catalyst itself. You need to have a very reactive catalyst. And in this case, we want to use a gold catalyst. And then you need the targeting unit here. This is very important too, to eliminate the off-targeting problem, because when you have a specific targeting, so you don't have to worry about the side effect about off-targeting problem. And then when you have a specific targeting unit, you need to make it as a complex, then you need to make it stable. And then whenever you inject this metalloenzyme here, and whenever you meet with your prodrug, and then they will react each other, and then this tumor cell will be tagged by the peptide, and then this peptide cause apoptosis inductions, and then the cell will adapt. And before we go for further analysis, we need to confirm first, is it really our metalloenzyme can be specifically delivered to the cancer? And we here prove by using in vivo imaging to prove the tumor-specific targeting by using HSA as a template, human serum albumin. And we modified with the RGD here. And this is the peptide, cyclic peptide, and we label with a size 7.5. And then we inject intravenously this RGD labeled HSA to intravenously to the nude mice, tumor-bearing mice here, and we observe, we took the image of the 6 hours and 8 hours. As you can see here in vivo imaging, so that's very clear accumulation in the tumor, while in other organs, almost no accumulations. Then after 24 hours post-injections, we sacrificed the mice and we took the major organs such as heart, lung, pancreas, femur, brain, muscle, and also tumor. And you can compare here that the highest accumulations is in tumor, and while in the other organ, almost no accumulation. So therefore, we can confirm that our metalloenzyme can be specifically accumulated in tumor. And we were very happy that our strategy might be possible to do in a specific area in a tumor. And here, when we have the specific organ targeting here with the metalloenzyme and we prepare the peptide 9, actually we prepare a lot of peptide at that time, but we choose the very appropriate peptide for this strategy. So here is the peptide 9, it's in active form when you attach the propargyl esters. And whenever they meet with the gold, then this peptide can be labeled on the surface of the cell and can induce cell apoptosis. And here's the cell viability analysis. As you can see here, peptide 9 in the presence of metalloenzyme, the cell viability is very low here. The cell growth can be inhibited. So this peptide 9 and metalloenzyme can be used as a starting material for therapeutic in vivo synthetic chemistry. And then we come to the in vivo therapy. So in this case, we want to treat our compounds. We want to use our compound to treat tumor-bearing mice. So first, we need to make tumor model mice here. We inject the nude mice with the SW620 tumor with the subcutaneous injections. And then when the tumor grows at the perfect size, we intravenously inject the gold complex. And then after some time, we inject with the peptide 9. And whenever they meet, we expect that the reactions occur in a cell, cancer cells. And during therapy, we observe the tumor growth, body weight, and also the survival rate. And here is a tumor growth during a therapy. As you know here, we did 10-time injections in consecutive days. And because we need more intense injections, as you can see here, that in gold only and peptide only, so a slightly cytotoxicity compared to the control group mice. And but when you use combinations between peptide 9 and gold metalloenzyme, the tumor growth can be totally suppressed. In this case, although they show, and for this group show slightly cytotoxicity, but it is really possible that to treat a cancer with our strategies, because it can totally inhibit tumor growth, and we are very happy that it might be promising for treating a cancer cells. And we also check the body weight. As you can see here, the body weight is relatively stable, but however, in the peptide 9 and in the presence of the metalloenzyme, so slightly decrease at the end of the treatment. But however, after the treatment, the body weight go back to the normal. So we can assume that it might be because of 10 times injections. It's really too much for injections. And but after treatment, the body weight go back to the normal. So we may say that it's really the side effect. We can neglect a little bit with the side effect. And we are very happy that this is an excellent result here. And we also check the survival rate of the treated mice. So here is the 3 groups: cell line and a peptide only and a metalloenzyme only did not show any significant difference. But when you treat the mice with a peptide 9 in the presence of metalloenzyme here, you can prolong the survival rate up to 2 weeks. And it's really excellent result that our strategy in vivo therapeutic chemistry can treat a cancer cells. But however, it's the disadvantage of these reactions and this therapy is that you need 10 times injections. It's a little too much, and it's not really able to be applied in a human or in a patients. So therefore, we need to improve our chemistry to get a much better reaction that can be used and applicable for treating cancer cells. So here we come up with a new strategy with this new SECD strategy. So in here, instead of gold, we were using a ruthenium as a catalyst, and we use alloc benzyl fluoride as a side chain to protect the drug active. And after that, we after removal the alloc moiety, and this is a resonance of the proton, and then you cleave the fluoride, and then you form a quinone methionine formations. And this structure is really reactive. So nucleophilic attack from amino acid, especially from hydroxyl and thiol, is really reactive against these structures. So then the cancer cells can be labeled on the surface by our drug or a peptide. And then we also did the same thing as in gold therapy. So here we also make a tumor model mice. We use SW620, and we also inject the subcutaneous injections on the shoulder mat of the mice. And then after the size, we have the perfect size, and then we treat the tumor-bearing mice with the ruthenium complex, and also after accumulated well in the tumor, we treat it with the peptide 9, and we also check the body weight, the tumor growth, and also the survival rate here. And during therapy, this special thing compared to the gold thing that we try only one injection, so we call it single injections. on the first day only. So here we have the 5 group of treated mice. Here, saline, peptide, catalyst, peptide and the catalyst, and also peptide and catalyst, but you doubled the dose. In the case of saline peptide catalyst, you did not any significant difference, because you can, in this case, it means that there is no activity in this compound, I mean saline peptide and catalyst. But when you treat a mice with a peptide in the presence of the metalloenzyme, you can inhibit the tumor growth significantly. But when you double the dose with peptide and catalyst, you can totally inhibit the tumor growth, even only one single injections. So therefore, we are very happy for this result. We can improve our chemistry from 10 injections to 1 injections. And this is a really excellent result, and even with a double dose. And then we'll also check the body weight. As you can see here, that the body weight is relatively stable, almost no different weight among the group, and we can confirm that almost no cytotoxicity were observed in this therapy. And then we also check the survival rate of the treated mice. As you can see here, saline, peptide, ILOC, and ruthenium almost no significant difference. Even ruthenium shows some slightly activity, but not so much different. But in the presence of peptide, ILOC, and ruthenium, you can prolong the survival rate up to 2 weeks. And when you double the concentrations of the dose, you can get like around 3 weeks prolonged for the survival rate. And we are very happy that actually this is also the first time that ruthenium can be used as reactions in vivo. And this is the first report that we report. And also maybe this is an excellent result, because we can confirm that ruthenium can be used for this strategy, and even more stable compared to the gold metalloenzyme. And then here is the images that we took on day 1 and day 8. And you can compare here a vehicle, I mean, this is saline only, you can see very significant difference, and you can see the peptide here also significant different, catalyst. But in the case of peptide, not so much different. And in the peptide catalyst, you can see in the double dose, there's no difference. So therefore, this strategy is really promising to be applied in cancer therapy. Yeah, I think that's all for my talk. Thank you very much for your kind attentions.